Hello, everyone. Thank you so much for joining us today. Welcome to the 2024 RUMEN Microbial Genomics webinar. I'm Milka Popova, and I'm here with Yanfen Cheng, who will be chairing today's session with me. So over the next 2 hours, we'll be diving into culture-dependent methods to study rumen bacteria, archaea, and protozoa. But before we get started, I just want to give you a couple of quick tips so we can get most out of this webinar. I, Yanfen, and any of our guest speakers are really happy to answer any questions you may have, so please feel free to post them in the Q&A box. And if you miss any of the webinars or have to leave early, don't worry, we'll email everyone a link to rewatch it later. So we are delighted to have with us Samantha Joan-Noel, an associate professor at the Department of Animal and Veterinary Science, Aarhus University. Her expertise lies in livestock gut microbiota with a strong background in microbiology, molecular biology, and bioinformatics. Dr. Noel's research is focused on strategies to mitigate the environmental impact on animal production. She is particularly interested in reducing greenhouse gas emissions from cattle, while simultaneously enhancing animal health and productivity. Samantha, the floor is yours.
Okay, thank you. And we will start by sharing. And start our slide. Okay, thank you very much for the introduction, Milka. I'm happy to be here. Thank you for the invitation. I've been asked to give a talk on bacteria isolation from the rumen. So I'm giving a talk titled Cultivating Success, the Progression of Rumen Bacterial Cultural Techniques. And I'd like to start with a little bit of history. So the history of bacterial cultivation from the rumen, it really took off in the 1940s, 50s with the research of Robert Hungate, who was a pioneer of rumen microbial research and developed the methods to culture anaerobic bacteria. In 1944, Robert Hungate isolated a cellulose degrading microorganism, and in 1950, he published the complete description of this technique. This method is known as the Hungate technique, and it's still in use today to isolate and to grow rumen bacteria. In short, this is where the culture media was boiled and then oxygen-free gas was bubbled through before dispensing it into rubber stopper tubes. A reducing agent was added to it to remove the remaining oxygen from the tube. And this was later refined to a roll tube technique where warm agar in the tube was rolled under cold water to form a thin layer of agar on the outside of the tube in which rumen bacteria could grow. During this time, there were different media compositions formulated and they were developed, and the isolation of rumen bacteria was possible. Moving further along, new technologies permitted the successful cultivation of rumen microorganisms on agar plates. These opened new avenues for the study of the physiology and the metabolic potential of many new microorganisms. Anaerobic jars that were airtight and had gas ports to exchange the atmosphere within with an oxygen-free gas mix were coming into use in the 60s, and these could be used in combination with gas packs to generate this oxygen-free atmosphere. And the Koi anaerobic chamber was developed in the 1970s, and this had an anaerobic atmosphere maintained within it, and the gas mix within typically contained 5% hydrogen, and this reacted with a palladium catalyst in the chamber, which could remove excess oxygen that would get in through the port and things that have been brought in. There are also rigid versions of anaerobic chambers made by a variety of companies that operate under similar principles. During the 70s and the 80s, many new bacteria were cultured from the rumen, and around this time at the end of the 80s, it was thought that maybe the main functional groups have been in culture. However, the explosion of culture-free technologies has proved this otherwise. So in the 2000s, the first widely available high-throughput sequencing machine became available, and that was the 454 pyrosequencer by Life Sciences. And culturing then took a bit of a backseat to these culture-independent methods as they took off. In 2005, for example, this FLX version of the 454 sequencer could generate 400 to 600 million base pairs per run. And since then, the technologies have only got more accurate, they've been faster, and they're cheaper per base. So the focus on rumen microbiology really shifted to culture-independent studies, as this eliminates the tedious and slow process of trying to grow anaerobic bacteria, and it gives a fuller picture of the community than was previously known from culturing studies. But it also meant that if you're not a specialized anaerobic microbiology lab, you can now study these less accessible environments via looking at their DNA. However, it did demonstrate that the rumen community was far more diverse than originally thought. And the culture-independent studies have really revealed gaps in our culture collections. And our knowledge on what is there has quickly expanded due to these culture-independent techniques. However, there are large gaps, and that means we have a limited ability to determine what the functional role of these uncultured bacteria are. If their databases, they were generated from the known bacteria, they underpin the comprehension of rumen metagenomic and metatranscriptomic data. Therefore, we need a more comprehensive reference genome collection. Furthermore, the composition of the culture collections do not represent the composition of the bacteria that's in the rumen. There is an overrepresentation of some taxa, like those found within the Lactospiraceae, and there's underrepresentation of other common taxa, like in the phylum Bacteroidetes. In recent years, there's been a renewed interest in culturing rumen bacteria. So why haven't we got everything yet? Maybe, are they just inculturable, or are we unable to sufficiently replicate the natural environment? Maybe they need interactions with other microbes that we're not taking into consideration. There could be competition in the growth media leading to the fast growers dominating the cultures, and then you'll be culturing the same thing over and over again. And perhaps it's just not enough attempts. I would like to now show some work that is going to address at least 3 of these points. So this is from an experiment that I've been involved in, where we were culturing rumen bacteria, but really focusing on the plant-adherent rumen bacteria community. So this experimental design here has an extra step where we're separating out the adherent community from the planktonic community. The samples were taken from rumen of dairy cows, and the solids were separated from the liquid by squeezing them through cheesecloth. The content was washed to remove the non-adherent cells and then blended to release the adherent cells. And this blended soup was serially diluted, and a series of close dilutions were then used to inoculate a series of liquid media in tubes. The same blended washed digester was then subjected to DNA extraction, and the total community from which the isolates came from was examined with amplicon sequencing of the V1 to V3 regions, of the 16S RNA. Then the identified isolates can then be compared to the original community for each sample. This cultivation approach was to use a medium that had been developed to better mimic the physical chemical conditions prevailing in the rumen in terms of the pH, the salts balance, and the concentrations. This was the RM02 medium, and to combine this with dilution theory to maximize the diversity of bacteria that was obtained. So in short, the aim was to dilute the inoculum until there is a high chance of inoculating a growth media with just one cell. This method is also referred to as the dilution to extinction method. And for this study, the optimum we chose was a theoretical 0.2 cells per tube of media. And how we got this number was by using dilution theory. So if you're looking at the graph on the left, you can appreciate it as the number of culturable cells that you inoculate into a series of tubes increases, so does the number of growth positive tubes in that series. But if you have 100% of your tubes that grow, then the chances are they were not inoculated with just one bacteria and are not pure cultures. So in order to achieve about a 90% incidence of growth positive cultures that are derived from single cells, the inoculum should contain an expected 0.2 cells. The number of tubes that will be growth positive in the series will be 20% of the total. And the number of tubes containing cultures derived from a single viable cell will be about 18% of the total. And this means you end up throwing a lot of tubes away, they have nothing growing in them, but you save a lot of time by having pure cultures from the start. And in practice, a series of close, in our case, fourfold dilutions were used to inoculate the series of tubes to encompass that theoretical 0.2 cell point. And we achieved a 0.1 to a 27 cell range. And the benefits of this approach is that there will be no bias towards bacteria that grow well in our chosen medium and will dominate in mixed cultures. We're also able to detect very poor growth in RM02 medium, as you can see from the picture here on the right. It is very clear and has a low color. It's just a pale straw color. And then also to get pure isolates from the initial inoculation. Again, you throw away a lot of ungrown tubes, but you save a lot of time in the purification steps. And that time and energy was then spent into getting more isolates. And overall, from the study, 6,680 tubes were inoculated. And this resulted in 1,037 isolates that were able to be subcultured. 828 of these isolates were able to identify with their V1 to V3 region of the 16S RNA gene, and they represented 658 unique sequence types. When these gene sequences were grouped together into operational taxonomic units at 96% similarity, they were 204 OTUs. 51% of them, 105 OTUs, represented potentially new species, and they were less than 96% similar to a previously cultured bacterium. 108 of these isolates were included in the Hungate 1000 project, and they went on to have their genome sequenced and added to a growing reference database of rumen bacteria. But looking at the success of the isolation, we can judge the success of the isolation by comparing the sequenced rumen community with the isolate sequences. And here 7.7% of the diversity, which I'm representing as OTUs, seen in the rumen sequences was isolated in the study. Out of the isolates that didn't cluster with a rumen community sequence, they appeared to be valid isolates, but they maybe represent the easy to culture but rare species. And 7.7% of the diversity shows that there is much further to go to capture the rumen diversity, even though around half of the diversity that we did culture seemed to be new. And to look at this a little bit further, we compared what was isolated in this study with the rumen community that it came from, and also what had been isolated previously and deposited in just one database, in this case, the RDP database. So all isolates that had been deposited in the RDP database at the time. And first of all, I'll note that the rumen community was not well covered by isolates that were already in the RDP database, with 90% of the rumen diversity not clustering with an RDP isolate. Then 75% of the isolate OTUs didn't cluster with an isolate from the RDP, so they represented new isolates found in the study. With this analysis, there is 103 OTUs representing previously cultured bacteria, and this number agrees with estimates of the previously cultured rumen species at the time. And then this is compared with the 169 OTUs that represent new isolates. It shows that more than 1.6 times the total number of the previously isolated rumen-associated species were gained in this study. There is, however, a large number to go, and this looks to be the tip of the iceberg in cataloguing the rumen community. And keeping with the iceberg metaphor, this is reminiscent of what's seen in the human gut microbiome culturomics study that demonstrates that maybe this isn't even the full picture. See if I can find a laser point here. So the number of species that could be seen here with culturomics has a very small overlap with the number of species that were seen by sequencing the community. There is also an overlap with the number of species from the human gut microbiome that was already known about. But what is suggested is that there is a potentially large unknown portion that's not seen with either of these methods. But back to the main study, we also wanted to see if we could increase the chances of isolating different bacteria by using a range of different media other than the main media of RM02. So alongside RM02 media, 9 other media with specific modifications were tested. Now, 5 of them were RM02 media with extra additions, and 2 of them were straight rumen fluid, straight clarified rumen fluid, plus 2 commonly used media to grow and maintain rumen microbes. And they were grouped together in a category known as, just called other media here. And isolates from all of the other media types are compared from isolates from the RM02 media type alone. A refraction analysis indicated that more diversity or OTUs was being sampled by using the combination of other media compared to RM02 media alone. However, refraction from the entire data set in the blue dot here for all media showed that the total diversity was not greater than both subsets of isolates, showing that RM02 media and the other media had isolates that were broadly similar organisms at the OTU level. And in this study, there was not a tremendously large increase of diversity by including the 9 other medias. There was a modest increase of 21.5% more OTUs that were gained by using the 9 other media. But contrast that to another study, another culturomics study, culture experiments done by Zahavi et al., where rumen fluid was taken and diluted and cultured on plates using a different media, medium N10, in a defined and an undefined form. So the defined form was with a VFA mix and the undefined form was with rumen fluid. These were anaerobically incubated. And then the entire microbial consortia that grew on the plate was scraped off and identified using 16S RNA amplicon sequencing and compared with the donor rumen fluid. And they found that 23% of the diversity could grow on the plates. And also the media that contained the rumen fluid cultured a greater diversity, almost double. But including the second media type increased the cultured diversity by 40%, which is greater than what was seen in the previous study. They also concluded that the largest influence on the diversity seen was the sample dilution, where the sample dilution reduced the competition in the abundant bacteria, and in the more dilute samples, it allowed the low abundant community members to grow. So the more dilute the inoculum, the more taxa was seen. And future perspectives for culturing rumen bacteria is heading towards the culturomics principles, where a high throughput cell culture aim to comprehensively identify species in complex samples like the rumen environment. And going forward, we need more high throughput methods to increase the number of isolates and therefore increase the diversity of isolates and reduce the gaps in the culture collection. We need to be thinking about screening thousands of cultures, not just hundreds of cultures, in order to statistically have a chance at getting some of those lower abundant members. And for this, equipment like the biotyper, the Smoldy biotyper, is quite essential because it dramatically speeds up one of the bottleneck points of isolate identification. Also, various forms of automation will be able to speed up this process. However, this is difficult as it has to be done anaerobically, which means robots in chambers. And the drawback to this is that these setups come at a high cost, and they're very specialized for isolating anaerobic bacteria, and they're not going to be accessible to everybody. But with high throughput culturing studies, the culturing conditions can be adjusted to increase the diversity of bacteria that you can isolate, with things to consider, including the donor material source, the dilution, the media formulations, and prolonged incubations. A limited number of culture conditions might be acceptable to, or okay to get the majority of the culturable bacteria. However, for the low abundant and the hard to get bacteria, a more targeted approach will be needed. And finally, the some interactions with other microbes within the rumen community could play an essential part for some bacterial species, and this is not addressed by the dilution to extinction techniques that I've presented. I'd like to thank you for spending time to listen to me today, and I guess we are able to answer some questions.
Thank you so much, Jim, for your nice presentation. And there were two questions in the Q&A box. So can you see, can you have a look at the box?
I'm going to stop sharing and see if I can find the box.
Yeah, and the first one is, what is the version of RDP database?
It is the version that is, I can't remember the exact version right now, but it was from the time that this was done, which is probably 10 years ago now. The 10 years old
ago.
version.
Okay, and the second one is from Rob. What gas composition do you use for this? 80% hydrogen is not usable in a lot of labs because of the risk of explosion.
I use, for the RM02 media, CO2. The 80% hydrogen is for growing methanogens. it's more important. And then in the chambers, there is a gas mix where you can use just 5% hydrogen.
Okay. And the third one, how can we relate functional metagenomics with culturomics?
I think that we can use information gained from the genomes assembled from culture-independent studies to direct how to culture particular groups. You can use that information to perhaps target something that is not in the culture collection and that you want. You can also use it to see where the gaps are and know what you are after. It also gives an idea of like how many are left, how large the uncultured space is left. I can see there's a question here about brands. Brands of media components. I haven't found that the brands are essential. There are some yeast extracts that are horrible to work with and some that are nice, but the salts and the more chemical based components, I haven't found brand differences, but I guess there are brand differences for some of the carbohydrates that you may add into the media. But I don't have any recommendations on the types of brands to use for making the culture media. It is quite specific. I can see a question saying, what is the best method to isolate methanogens? And I'm going to let Mela tell you about that because she's on methanogens next.
Yeah.
Is it possible to standardize the isolation procedure? I'm not sure if I know what you mean by standardize. When I have done it from different animals, I have used a standard procedure all the way through with the dilutions, finding the point to find that 0.2 cells. Once I'd found it for one animal, it was quite reproducible. Every time we took a rumen sample, we would be able to hit it in a reproducible way. But I'm not sure you would want to totally standardize isolation procedures because the slight differences might mean that you capture more diversity when different people try them. Okay, there's a question saying, why use CO2 over something like 80% M2, 15% CO2, and 2.5% H2? The rumen culturomics labs seems to like high CO2, but I'm not sure why. The RM02 media that I was describing is designed to be buffered with CO2. If you use a lower percentage, then you don't have the same pH as if you're bubbling with 100% CO2. I have actually accidentally done that, and I was wondering why my media went cloudy when it came out of the autoclave, and it turned out that there was something coming out of solution because the pH wasn't right, because I'd used a gas mix rather than using the straight CO2. However, for the other medias, you can use different gas mixes. It depends on the type of buffering system that they're designed to be made with. And there's another question saying, did you use any kind of antibiotics to negate the typical or common organisms to overgrow? I didn't, but you could. And it would be particularly useful if you're isolating pathogens to use antibiotics. I wanted to see if we could, without interfering, just get good diversity by getting higher numbers. In your experience, rumen fluid quality an important reagent? I know that where you source your rumen fluid from affects how easy it is to work with it in media, because it needs to be filter sterilized, and that cows that are fed forages, high forages and hay, will make it easier to be able to process it, to use it in media and have a very clear media, whereas cows that have been fed concentrates make a more cloudy rumen fluid, and it's quite difficult to get through a filter at that stage. So in terms of quality, that is important, but I don't know how you would identify a bad quality cow to get rumen fluid from. Okay, any recommendations for isolating co-cultures, microbes that are dependent on another? And I've been thinking about that, is to, rather than go to dilution to the extreme point, you could dilute so that you are inoculating with a smaller number of cells, but greater than one. So to try and get these reduced communities, to see if you could to co-culture them with something else. If we culture the bacteria in vitro in fluid, will we lose the solid associated bacteria? If you culture a bacteria that is associated with the solid fraction in liquid, it will still be able to grow in liquid. It should still be able to grow in liquid, or many of them can. I'm not sure I understand that question completely. Any recommendation for transportation of rumen content samples from a fistulated animal to the laboratory? I use the method. We transport them in coffee thermoses that if you add a large volume and it's a short trip, it will remain anaerobic because of the volume. If you have small volumes, they are prone to be get oxygenated. So if you want to transport rumen content, then transport a big bucket of it. On the question of rumen fluid, is there a standard diet for cattle used to collect rumen fluid from? Is changing the diet a good way to increase culture diversity? I would say there's no standard diet if you're looking to isolate bacteria. The standard diet is good for if you want to use the rumen fluid for in media. So if you want easy to handle in the lab rumen fluid, but it's a good idea to use different diets to culture different diversity for the inoculum, the source of culturing the bacteria. What is the incubation period for the dilution method? I left them to grow for 2 weeks so that if they're extremely poor growers, you give them a chance. And you're also, I was aiming to add one cell in, so it might take quite some time if it's a poor grower in the media to see that it had grown. However, the rumen bacteria typically grow faster than that. Do you recommend some conservation media too long? Oh, so if you want to store rumen fluid to get isolates from, I would recommend adding some glycerol, some anaerobic glycerol into a Hungate tube containing your anaerobic rumen fluid, and then store them in -80. So I think maybe 30% glycerol total, and then store your rumen fluid at -80, and then you can try and culture from it at a later point.
Thank you again for Sam, and it's the time for our next presentation, and thank you again for your next presentation. So if our audience, if they have any further questions, they can contact you later, okay? Now it's time for our next presentation, next speaker. Mala, are you here? Could you please share your screen?
Okay.
Yeah. So Mala got her PhD. Got her PhD in New Zealand, and then she moved to France, to Indra, to did her postdoctoral for 2 years, and then she went to Belgium, to Ghent University as a researcher. Now she's a researcher at Ghent University, and today she will introduce how to isolate rumen methanogens. So please welcome.
Thank you, Yanfen. Can you see my slides?
Yes.
Okay. So hello, everyone. In this presentation, I'm going to share my experience on isolation of rumen methanogens using dilution to extinction technique. Some of the information may overlap from the previous presentation because the methods and techniques are more or less similar for bacteria and archaea. When consider the methanogens, they are belong to archaea. In the rumen, methanogens are the major archaeal group present in the rumen, although they contribute around 0.3 to 3% of prokaryotes. They perform an important job, that is removing the hydrogen produced by the feed fermentation. For the effective fermentation, this hydrogen needs to be removed. That is done by these rumen methanogens. They are strictly anaerobic. And when consider the rumen methanogens, there are two major groups, hydrogenotrophs and metallotrophs. Major group is hydrogenotrophs. More than 75% of rumen methanogens belongs to this group. They use hydrogen to reduce carbon dioxide to produce methane. The dominant genus in this group, from this group, is Methanobrevibacter. The other important group is metallotrophs. They consist around 10 to 16% of the rumen methanogens. They use hydrogen to reduce methanol or methylamine to produce methane. The Methanosphaera and Methanomycelicoccus are two important groups from these metallotrophs. So molecular-based studies gave a lot of information about the rumen methanogens, but to understand the metabolic and physiological properties of methanogens, we need to have the pure cultures. So when consider the culturing, the two important things we need to think is culture techniques and media used. So roll tube method is the popular method used to isolate methanogens. So you can use Hunger tubes or serum vials to this technique. But sometimes picking the colonies from these roll tubes is a bit difficult. So researchers also use pour plate method, agar plates, by using anaerobic chamber or anaerobic jar. The media used is another important things need to be considered when consider the culturing rumen methanogens. There are solid and liquid media are used to isolate methanogens. This media should provide essential nutrients as well as specific growth factors such as coenzyme M, vitamin B, etc. So sometimes some of the methanogens need some growth factors. We don't know what it is. So that's the reason most of these media consist rumen fluid. It can vary from 5 to 30%. When considering isolating method, the popular method is dilution to extinction techniques. This was explained in the previous presentation, where the initial inocula is diluted until the tubes contain less than 1 cells per tube. Nowadays, the single cell encapsulation method also becoming popular. Another important technique is co-culture method, where hydrogen-producing bacteria is used to provide hydrogen needed for these methanogens. For the slow-growing methanogens, this one is suitable because many rice culture clade methanogens are isolated by this method. And there are other methods such as diffusion, chamber method, etc. But in this presentation, I'm going to share the dilution extinction technique I used to isolate methanogens. So what is dilution to extinction technique? Where the sample is serially diluted to the extent that each final tube contains a single or no microbial cells, less than 1 cells per tube. And by this method, Kenter et al. Isolated about 60 rumen bacterial species. So we slightly modified this method to isolate the rumen methanogens. So we performed several experiments. In the first experiment, we wanted to know what is the suitable media and the blending time. For this experiment, we used adult sheep as an inoculum donor. So prior to this experiment, we also estimated the methanogen concentration in this sheep. So we counted the fluorescence cells under the epifluorescence microscopy, and also counted the DEPI-stained bacterial cells or prokaryotic cells. But counting fluorescent cells to estimate the methanogens sometimes underestimate the total methanogens, because it's known that the methanomycelicoccus is a lack of the F420 fluorescence cofactor. So we collected the rumen fluid. So we estimated the methanogen concentration is around 10 to the power 8 cells per milliliter. Then we collected rumen content from this sheep, and 40 grams of rumen content consist both solid and liquid were blended with 360 milliliter of RM02 base. So when consider the blending, we had 2 blending time, short and long. In short, we set the blender in high setting and we blended for 20 seconds. In the long blending time, we blended 4 times 20 seconds. And between each blending, we had 60-second resting time. And we used two different media to check which media is suitable for the methanogens. So BY plus media and RM02 media. So the blended inocula was diluted into RM02 serum vials containing RM02 base. This initial diluted inocula were further diluted until it has 10 to the power minus 4 cells per milliliter liquid. And each of these diluted inocula were inoculated again into the Hungate tubes containing either BY plus media or RM02 media. So 0.5 milliliter of this diluted inocula was inoculated into these Hungate tubes. So as a substrate, we used methanogens substrates, that is sodium formate, sodium acetate, methanol, and hydrogen carbon dioxide mixture. And we incubated for 30 days, and we checked the methane production in the headspace by using gas chromatography. And after the initial experiment, we estimated the most probable number, and we found out viable cell counts were greater in BY media compared to the RM02 media. But when consider the blending time in BY media, we didn't see much difference between long and short blending time. But in RM02 media, the short blending time gave more viable cells compared to the long blending cell. We repeated the same experiment again by using the same setup with slight modification. So when we repeated the experiment, we only used the long blending time, but with 2 different media. And the dilution factor is the same, but we diluted until 10 to the power minus 1 cells per milliliter. And instead of 5 tubes, we incubated 10 tubes per dilution. One of our objective was to isolate methanomesylicoccus also. So we wanted to know which rumen media is suitable for this group of methanogens. The substrate mixture was the same, and incubation duration reduced to 21 days. And we checked methane formation in the headspace, and also we checked the terminally positive methane positive tubes under the microscope for the fluorescent cells. We know methanomesylicoccus doesn't have the cofactor. So we also did for the selected tube qPCR with the archaeal as well as methanomesylicoccus specific primers. After first 2 experiments, we found out the viable cell counts are greater in BY plus media, so we choose to use this media. And when we consider the BY plus media, we didn't see much difference between the blending time. And also we found out that RCC or methanomesylicoccus only presents in the BY plus media. So we decided to use this media for our future isolation experiments. So we started our real isolation techniques or experiments. The setup was the same. We used the same sheet, and the modification were we only used long blending time and only 1 media, BY plus media. And also we reduced the dilution factor from 10 times to 4 times. And we, instead of 0.5 milliliter, we use 0.1 milliliter diluted inocula to inoculate the Hungate tubes containing methanogen substrate. We checked the methane formation in the headspace, and also microscopy. We checked the fluorescent cells, and also we did the PCR and sequenced to check whether which methanogens are present in the terminally methane positive tubes. And during this experiment, we could isolate, or we could get a tube containing ISO3F5 that belongs to the Methanosphaera species. We repeated the same experiment, but this time with a short blending time with a BY plus media. And instead of 10 tubes, we also increase the number of tubes that is inoculated per dilution. So we increased to 60 to 100 tubes per dilution. In addition to the substrate mixture, we also added the cofactor coenzyme M. And the incubation duration was the same, 21 days. And we checked the methane formation, microscopy were used, and also we performed the PCR to check which methanogens are present. And during this isolation experiment 4 and 5, we could isolate 5 methanogens. So I will briefly go through the isolated methanogens. So we have to remember the techniques, the dilution extinction techniques. We didn't use antibiotics in any of these steps. So the tubes which contain the single methanogen species also contained the bacteria. So the next step was to purify these methanogens from the bacteria. So for this, we used several techniques. The major one is the dilution that we did this time with the antibiotics. So as antibiotics, we used combination of streptomycin, ampicillin, vancomycin, and bacitracin. And also we use lysozyme. In some cases, we had to use the heat treatment. So we place the tubes containing methanogens and bacteria in the water bath at 50 to 55 degrees Celsius for about 10, 20, or sometimes 30 minutes. The purification of ISO3F5 methanosphaera was possible during this heat treatment. It's also possible to use or inoculate the terminal methane positive tubes into rolled tubes and pick up the colonies. But in our method, we didn't use rolled tubes. And once we know they are pure, then we have to check the purity by using several methods. We test whether they are pure. The first one is we checked our cultures under the phase contrast and epifluorescence microscopy for cell morphology and fluorescence. And again, methanomycelicoccales, for them, it's not possible. And also, we grow the purified culture with the bacterial substrate, that is mixed sugar, peptone, and yeast extract solution, both aerobically and anaerobically, to be sure there is no aerobic or anaerobic bacteria present. And also we perform the bacterial and archaeal PCR and sequence them. Once we know they are pure, we can perform full or partial characterization of these isolated methanogens. We mostly perform the partial characterization of these isolated methanogens, such as substrate requirement. So by including, omitting methanogen substrate, you can check the substrate requirement. And also we check the growth factor requirement, whether yeast extract, rumen fluid, vitamin solution, or coenzyme M, whether they need it or not, or whether they increase the growth, etc. So you can check that by omitting and including these growth factors. And also we checked optimum temperature and pH and gram stain, and also some of them was genome sequencing of some of them was performed. So some of the isolated methanogens, I briefly go through it. So the first one we isolated was ISO3F5, that is a Methanosphaera species, and we found out they only grow when methanol, acetate, and hydrogen CO2 is present, and they require yeast extract. So it's maybe the vitamin B requirement is important for them. And the genome sequence of this and partial characterization of this methanogen is already available. And the other two methanogens, the genome sequenced were ISO4G1 and ISO4H5, which were isolated at the fourth isolation experiment. They are methanomycelicoccales, and they have the hydrogen-dependent methylotrophic methanogenesis, where a methanol and methylamine is used. And we also found out they are lack a coenzyme F420. And again, the genome sequence and partial characterization of these two isolates are already available. See, the other three isolates, we didn't perform the genome sequence is not yet available, but one of them is belong to methanomycelicoccus. It's again, it has the same substrate requirement like the other two methanomycelicoccus, methanol and methylamine. And also we had two isolate from Methanobrevibacter species. So these 6 isolates were isolated during my experiment. And this dilution to extinction technique was used by the other researchers in other environment, and they also succeeded isolating these rumen methanogens. Recently, the Shi et al. Reported that they isolated 2 Methanomesylicoccus culture by using this technique, and also Hood et al. They also isolated 2 rumen Methanosphaera cultures by this method. But for them, they use antibiotic during the initial dilution to extinction technique, but we didn't use then antibiotics during this process. And also in these 2 studies, they use roll tube to culture the terminal methane positive tubes during the purification. So although this is popular and most used technique, there are some limitations on this technique. For example, the low abundance microbes, it's not suitable for them, because when you dilute to reach the less than 1 cells per milliliter, you may lose the low abundance methanogens. And also we have to work with this number of thousands of tubes, so it's labor intensive, time consuming, and also resource intensive method. Maybe it's not suitable for slow growing methanogens because we are giving high concentration of substrates and hydrogen. Maybe it's not suitable for the slow growing methanogens. And also when we dilute until the less than 1 cells for archaea, you can not eliminate all the bacteria at that dilution level, so we will end up with mixed cultures. So that is end of my presentation. At this opportunity, I would thank the PGGRC who are the funding, who gave the fund for this research, and also the RUMEN microbiology team at Ag Research for their support during this isolation experiment. Thank you.
Thank you, Melar. Thank you very much for this very nice presentation. There are already some questions in the Q&R box. I don't know if you can see it. I'll read the first one because I have a similar question as well. So it's a question from Rene, who is asking, what is a diffusion chamber method? And I wanted to ask you, okay, go.
Diffusion chamber method, that's similar to you incubate the methanogens or in their natural environment. But I don't know whether this is used for the methanogens, but for the bacteria that is used.
Okay. And I had a similar question. How do you use co-culture for isolation of methanogens?
So that is actually used by to isolate the rice culture plate. So they are, instead of using hydrogen, they use bacteria that provide hydrogen as a source for the hydrogens.
So you just apply bacteria in your
yes, that
isolation
particular
tubes.
Bacteria, you will know which bacteria present there because so the known bacteria is used to provide hydrogen. For the slow-growing methanogens, I saw some of the articles by using this method, they could isolate slow-growing methanogens.
The next question is, do you find methanogens belonging to genus Methanothermobacter?
I don't think they are belong to, there are methanothermobacter present in the rumen. I'm not sure.
So another question is about the dilution factor. Why is the dilution factor changing from power 10 to power 4 in experiment 3, as well as the mLs from 0.5 to 0.1? And how do you get the cofactor? How do you add the cofactor?
Cofactor was added, we filter sterilized the cofactor, and then we added after we autoclave the media into the main media, because you can get the coenzyme M. You can buy it and we can filter sterilize them.
Okay. And for
So
the
dilution factor, we thought we didn't have a lot of tubes which has the methane positive, so we reduced these. The first two experiment, we didn't have a lot of terminal methane positive tubes, so we reduced it to dilution factor 4, and after that we reduced the inocula to 0.1 instead of 0.5 to based on the MPN calculation.
Another question is, which archival primer do you use for identification?
We use, I think it's 9 on V4 region, that is 960-35 and 1368. The reverse primer is 1368 and the forward primer is around 915.
They should be in the paper anyway.
Yeah, I think so.
Yeah. And then you've been asked to suggest a method for long-term preservation of methanogens, as glycerol doesn't work that well.
Yeah, we always use the 5% DMSO to preserve the methanogens.
And you never store them alone without the preservation?
No, we always use the 5% of DMSO to preserve for long term. So we prepared the media with the 5% of DMSO and we preserve like that.
Okay. Can you store rumen content and then use later for culturing methanogens?
I didn't do it, but I think it's possible. You can preserve them with glycerol and use them to isolate methanogens, or preserve them with DMSO and use them to isolate methanogens. It's possible, I think.
What is the shelf life of the culture medium once prepared?
We used it within 6 months. It's always stored in dark. So we prepare it, autoclave it, and kept it dark till we use. But if it is more than 6 months, we don't use it.
So there are no more questions in the Q&R, at least I don't see them, but I have another one. Do you have some recommendations depending on the type of methanogen we are targeting? I mean, methylotrophs or hydrogenotrophs. Is there something that works?
I think you can use the initial enrichment with the targeted group, because if you are going to targeting the methylotrophs, you can use only the substrate for the methylotrophs. Yeah, but it also can grow, but I think it also increase the chance of methylotrophs. Yeah, you can enrich them using the substrates that is used by that particular methanogens. Okay. Before you go for the dilution to extinction technique.
Okay. So there are no more questions. Thank you very much, Madar.
Okay,
And we're going.
I stop sharing or it's still
Yeah,
shared?
please. Yeah.
Okay.
So we'll proceed to our next speaker, Tim Hackman, who is associate professor at University of California, Davis. His research interests include improving nutrition of ruminants through study of the microbes of the rumen, increasing supply of microbial protein available to animal digestion, and discovering biochemical pathways that microbes use to ferment carbohydrates in feed. His lab cultures bacteria, methanogens, fungi, and he's going to talk today about protozoa.
Okay, well, thank you for that introduction. Thank you for inviting me to speak. And I'll talk about culture techniques for ciliate protozoa from the rumen, recent advances, as well as persistent challenges. So ciliate protozoa are key microbes in the rumen. They were first discovered in the rumen over 180 years ago. These were the first microbes that were found in rumen contents, and they've fascinated microbiologists ever since. I have a video to play, but it doesn't seem like it's playing. Here we go. All right, so we have rumen protozoa here swimming around in rumen fluid.
I don't see the presentation. I'm sorry.
You don't see the presentation.
No.
Let me stop the share, and thank you for pointing that out.
Okay.
Can you see it
It's
now?
working. Yes.
Okay, great. Okay, thank you for stopping me before we went too far. So ciliate protozoa were discovered over 180 years ago. They fascinated people ever since. And here's just a video of the rumen protozoa under the microscope. This is mixed rumen fluid, but what we see first are the protozoa. There are bacteria that are visible here, but they look like little dust particles compared to the protozoa. So this is what we notice first. So ciliate protozoa are key microbes in the rumen. They have both good and bad roles. One of their good roles is that they're armed with carbohydrate active enzymes. And as a result, they can digest feed. A way to show how important they are, we can look under the microscope. We can see them consuming feed particles, but a way to quantify this is that we can remove them from the rumen. You can remove them using chemicals. And if you remove them, it'll lower fiber NDF digestibility by about 20%. So this gives an indication that they really are contributing to digestion of feed in the rumen, and that can't be compensated by the presence of bacteria. But in addition to having these good roles, they have bad ones. One of the bad roles is that they act as predators of other microbes. So you can see this protozoan here swallowing this fungus. They eat bacteria, they eat protozoa. Basically anything that fits in their mouth, they will eat. As a result, they will decrease the amount of microbes that flow to the duodenum and are available to animal digestion. And if you remove protozoa from the rumen, it'll increase microbial protein flowing to the duodenum by about 30%. So we have both these good roles and bad roles, and it's still debated whether they are a net benefit or a detriment to the animal. Really what's going to help solve when they're a benefit or when they're a detriment, I think, is culture. Rumen. So I'll go over 3 different techniques that can be used for culturing. We can use ex vivo cultures, we can use in vitro batch cultures, and we can use in vitro continuous cultures. So I'll start with ex vivo cultures where we use protozoa from fresh rumen contents. What we're doing is we're taking rumen contents from the animal. Then we separate protozoa from the bacteria. We see that we have a mixture of protozoa and bacteria here. We'll separate them usually on a filter cloth. I'll get into that. And once we have separated cells, we can use them for metabolic experiments or anything that we want. The technique that we use for separation is key to success. So early approaches attempted to separate cells with varying degrees of success. They typically use sedimentation or centrifugation. So you can put rumen fluid in a separatory funnel and you'll see protozoa settle down to the bottom. You can use centrifugation to try to pellet the protozoa before the bacteria. We saw how big the protozoa are, so there was a thought that this would help separate them. But it's found that this leads to really high contamination. There was a study that measured the contamination, and it found that almost half of the protozoal nitrogen in one of these separated samples was actually bacterial. So you had a lot of protozoa, but you also had a lot of bacteria. More recent approaches have used filtration through a cloth. This was introduced back in the 1980s, but it's been refined over the years, and this is the technique that we use. So what you're doing is you're putting a cloth on this funnel. The cloth has pores that are big enough for bacteria to pass through, but small enough that most protozoa are retained, and then you wash them. And this technique leads to low degrees of contamination. When we've measured it in our experiments, it's only 0.2 to 0.8% contamination that we have. So it's almost all protozoa, just a small amount of bacteria or methanogens. And just to show you what you would typically get, we have here on the left our starting point rumen fluid. We have protozoa definitely, but we have a lot of bacteria. We have feed particles, which are covered with bacteria. Methanogens are in there, fungi as well. In the middle, we have what we call particle-free rumen fluid. Here we've removed feed particles, and it looks a little bit cleaner. And then here on the right, we have our protozoal suspension, which is what we've achieved after washing. And you can see that it's quite clear. There are some bacteria that you can see, but for the most part, this is a clean sample. And we can use it for metabolic experiments. So these ex vivo cultures have many different applications. In one of our experiments, we looked at how we metabolize, or how protozoa metabolize glucose, and we found that they metabolize glucose much faster than bacteria. So we have here on the right, protozoa metabolizing glucose much faster. Reserve carbohydrate, they're synthesizing it much more quickly than bacteria. Lactate, surprisingly, they're producing a lot faster than bacteria. Propionate, they produce much slower than bacteria. It's not much of a surprise that they are taking up glucose faster. That was thought to be the case, but it is surprising that they're producing a ton of lactate. It was thought that protozoa will moderate fermentation, that they'll take up glucose quickly, but then they won't ferment it, but we're showing that they are producing a ton of lactate. Other people have done other experiments where they show that protozoa can respire with oxygen. There's another experiment showing that they can consume lactate. So if you incubate them with lactate, they will use the lactate. In our particular experiment, when we're incubating with glucose, we're seeing that they're net producers of lactate. So it all depends on the conditions that you're using. And they have applications beyond just metabolic experiments. So since you get this relatively pure preparation, they've been used to prepare cell extracts to measure enzyme activities. They've been used for standards for PCR and also samples for chemical analysis. If you just want to know what protozoa are made up of, this is the technique that you can use. So this was recently used to determine the amino acid content of ciliate protozoa. There are definite weaknesses. Is that only short experiments, minutes to hours, are possible. So long periods will cause protozoal death and bacterial overgrowth. So just to give an idea of how bad this can be, our experiments lasted 80 minutes. And by the end of that 80-minute experiment, bacterial contamination already increased 50% over the starting value. Protozoa will die. We've tried to carry these cultures overnight, and when we get there in the morning, there are a couple of protozoa swimming around, but it's almost all bacteria. So we're really limited in what we can do. Okay, so that's ex vivo cultures. What I'll cover next are in vitro batch cultures. Here we're growing protozoa in tubes, and this is probably most similar to the cultures that we've seen with bacteria and methanogens in the earlier talks. What we're doing is we're taking a single protozoal cell from rumen contents. We're inoculating a tube of media. We're letting the protozoa grow. And then a couple of days later, we're going to transfer the cells to fresh media. The proper care of these cultures is absolutely key. So these cultures have to be fed a solid diet, and they have to be fed it daily. You have to feed them a diet of typically grass and wheat. And if you don't do this, they will quickly die. It's even more complicated. The amount of feed that you have to give them is carefully calibrated. You have to do it to keep the protozoa alive, but prevent overgrowth of bacteria that are also in these cultures. So you have to feed them daily. And another difficulty is that you can cryopreserve them, but only for short periods. The longest period that I've seen recently reported is 3 months. So this makes it a bit difficult to preserve these long term. It's not like with bacteria where you can just grow a culture, keep it in the freezer for years, revive it later. It can be quite challenging. But if you put in the time, then it is technically possible to maintain them for years or for decades. It's just a lot of work to keep them going. Another weakness is that there are bacteria that are present. So I've said that we start with a single protozoal cell, but we also have bacteria that are present when we start these cultures. And this is not a mistake. You have to do this. Bacteria have to be present. You can eliminate the bacteria with antibiotics, but then protozoa die within days or weeks. Multiple groups have shown this. The only way you can stop them from dying after giving them antibiotics is to feed them live bacteria again. Feeding them dead bacteria, for example, won't do it. Feeding them growth factors, whatever growth factors we can imagine, it's not able to do it. And because these bacteria are present, it is an issue. The cultures contain only about 20% protozoa by mass. So you do have this single species, this clonal culture of protozoa, but it is actually more bacteria than it is protozoa. So this makes inferences challenging to make. They still have useful applications. They can be used to prepare cell extracts to measure enzyme activities. They're used to measure growth rate. There's an experiment that shows that monensin slows their growth, and that helps explain why monensin can change populations of protozoa in the rumen. I'll end with in vitro continuous cultures. So with in vitro continuous cultures, we are growing protozoa in fermenters. We are collecting rumen contents and then inoculating a fermenter. We have a fermenter here at the right. We provide this fermenter with feed. We provide it with a continuous supply of buffer. So we have this arrow here, the buffer's entering through this line, and we have continuous removal of spent culture. We can follow this line right here. The proper design of these fermenters is key to their success. If you just have standard fermenters that you use for growing rumen bacteria or for any other bacteria, it's not going to do the job. You have to include a mechanism to physically retain the protozoa. So people will oftentimes include a bag of feed that will give a spot for the protozoa to sequester. Another technique is just to stir these slowly. So you can see the stirring mechanism here. If you turn it down, then feed particles will settle down at the bottom of the fermenter, and that will give a habitat where the protozoa can survive. So you have to be able to give them a way to be retained in the fermenter. Otherwise, they're washed out very quickly within a few days. If you do this maintenance properly, it is still possible to maintain them for weeks. You can keep them going for a while, maybe even longer than weeks. It's so much work involved. I don't think that people have maintained them longer than that. One caveat, though, is even with this proper maintenance, the protozoal populations are still about 10-fold lower than in the rumen. In the rumen, the concentrations are about 10 to the 5th to 10 to the 6th per milliliter. In fermenters, they're only about 10 to the 4th to 10 to the 5th. Bacteria and labor are definite weaknesses. So these cultures contain only about 10% protozoa by mass. So we're really struggling to maintain them. And they require a lot of work to maintain. So a lot of papers will describe their system for retaining protozoa. They'll characterize it, but then they won't go much beyond that. What's happening is that they are limited by the amount of work that they have to go into maintaining these things, and it limits the time for studying the protozoa themselves. If we could use them more frequently, it would be a good complement for in vivo studies. So in fermenters, a good thing that we can do is we can remove protozoa, and we can do it without using toxic chemicals. In the animal, we can remove protozoa, we can defaunate animals, but this requires toxic chemicals, and it's difficult. We don't know what effect it has on the microbial population. Here, you can just let them wash out. And so it provides a really good opportunity to compare a fermentation with or without protozoa. But the few studies that have tried to study protozoal biology show inconsistent results. For example, removing protozoa decreased feed digestion in one study. This is what you would expect based on in vivo studies. But in another study, it increased it. And it's not really clear why we see these inconsistent results. I think we just need to do more of these studies. We also need to be more critical about how we're designing these and whether we're affecting things like feed digestibility in the way that we design the fermenters. There are definitely challenges to be overcome. Culturing protozoa in the absence of bacteria is a must. So we're not going to be able to create cultures like we have with bacteria and methanogens until we can figure out how to culture protozoa free of other organisms. One major hurdle is we need to determine why protozoa require bacteria in the first place. We know that when we eliminate them with antibiotics that protozoa will die, but why does that happen? Are antibiotics toxic to protozoa? It's been suggested that that's the case. I think it's more complicated than that, but we really don't know. Another hurdle that we have to overcome is making methods more repeatable. Some of these techniques I've found are more art than science. So I've described in very general terms how to accomplish these, but figuring out how to do this yourself in the lab is a real challenge. You find out that there are tons of variables that have to be controlled. And towards this end, standardized protocols could help. So we now have a detailed protocol for ex vivo cultures. This paper was accepted last week. I'll provide a link at the end of the presentation. And it goes over how to collect rumen fluid, set up the rumen fluid in the lab, prepare the particle-free rumen fluid, prepare the filtration assembly, transfer particle-free fluid, wash the fluid, retrieve protozoa, and prepare the protozoal suspension. We go over in detail all the steps. We even have photos to help show what you would expect to see. And we have images of protozoa during this process. So I know I've listed a lot of limitations, but I think overall research with protozoa has a bright future. There are several techniques for culture available. Progress is being made in making these techniques more repeatable, and a role for protozoa can be better determined, and populations can be better manipulated from what we find from these cultures. I'd like to acknowledge the co-authors on the paper that I presented at the end, Arup Sen here at UC Davis and Jeffrey Firkins at Ohio State. He has a lot more experience than I have. I'd like to acknowledge funding that I have from the USDA and Department of Energy. And with that, I have some links for the ex vivo protocol and other papers published by my lab. The ex vivo protocol is at the top. And then I also have a link to our lab manual. So if you're asking, how do we make media? What chemicals do we use? That sort of thing. Everything is outlined in this lab manual. And this covers culture of protozoa, but it also covers culture of bacteria and methanogens. So I'd like to thank you for your attention, and I'm happy to answer questions that are in the chat and otherwise. So it looks like that the first question is, what is the pore size of the cloth? So when we do it, it is 20 microns. You can choose different pore sizes if you want to isolate different kinds of protozoa. 10 microns is another common one that will result in loss of fewer of the small protozoa. You can go up to 50 or 100 microns if you want to isolate larger protozoa. So again, our protocol is 20 microns. and you can see what cloth that we use. That's important too. But you have flexibility. Okay, and there's a question about the talks tomorrow that I think the others can answer better than I can.
Yes, they will be recorded.
Okay, yeah. Do specific bacterial species need to be present to sustain protozoa in vitro? That is a good question. There is one paper from Japan back in the 1980s that took pure cultures of bacteria and used them to sustain these protozoa. They eliminated all the other live bacteria and used these pure cultures. I think one of them was E. coli. I can't remember exactly. I don't remember exactly what they found and what they suggested, and if what exact species needed to be present. But this is an interesting question, and one way that we could potentially overcome this hurdle of, I can get to it. There. Culturing protozoa in absence of bacteria is just to replace the mixed bacteria that we have with cultures that we know very well. Okay. The next question is in between the halotrix and the antidinomorphids, which are more difficult to grow and maintain. It depends on the culture technique that you're using. For ex vivo cultures, it's very easy to isolate them both. I think I lost the question there, but it's easy to isolate them both. For in vitro batch cultures, it's comparatively easy to culture the entadenomorphids. It is very hard to culture the isotrichids. That's what I'm going to call them. Hungate back in the 1960s, I think, cultured them, but that's the only paper that describes it. And then in vitro continuous cultures, I'd have to look at the data. I think you can definitely culture them both, but I can't comment on which one is easier. Okay, so cryopreserved protozoa samples from the EU project were revived after 20 years in liquid nitrogen. That is great to know. I was not aware that work was, or those protozoa were still in the world. I thought that they had been lost. So it's great to know that you can cryopreserve them for that length of time. You could probably comment more on the protocol that was used. It was much more involved than the more recent protocols that are used to cryopreserve them for only 3 months. So that is great news that you can cryopreserve them longer. So the next question is, several diseases causing protozoa are cultured independently. If we can determine the pathogenicity, maybe that could be possible. So I think that there are a couple of things to unpack here. So if the question is suggesting that protozoa in the rumen are pathogenic, I'm not sure that that's been demonstrated. They're commensals. There have been protozoa from other environments that have been cultured without bacteria or methanogens. They could include pathogenic protozoa. It's not been solved with rumen protozoa yet. That's all I'll say, is that there's been a lot of attempts to culture them without bacteria and methanogens, and it's not been solved yet. So it's an open challenge. What benefits will protozoa have in improving the utilization of low-quality feeds? I think the major thing is that they digest fiber. So if you remove them from the rumen, you decrease fiber digestibility by 20%. So that gives an indication of how important they are. Is there an explanation for why the number of protozoa in continuous culture system is lower than in the rumen? Could it be due to a lack of an attachment matrix such as rumen-villi fibrous matrix in the rumen? It's a good question why we're not able to maintain higher concentrations. This is just the best that we can do. So yeah, we put bags of feed in the fermenters. We slow the stirring down. There have been a couple of other things that people have tried. They've tried to put sponges in fermenters. This is the best that we can do. It's just challenging to keep them around. How long can we feed the protozoa to preserve them? So if you feed them every day, then you can maintain them for years or for decades. I guess one other detail to add is that there is a protocol that will allow you to feed them only 5 days a week rather than 7, so you can at least take the weekend off. I don't know how long you can maintain them with that. I've done it for a couple of months. I haven't tried to do it for years or for decades. So you can definitely feed them and preserve them for a long time, but it's just a lot of work. And then the last question is a link to the procedure for cryopreserving the rumen ciliates, the ones that have allowed protozoa to be cryopreserved for 2 decades. So that is good to have that link there.
May I ask one last question?
Yes, definitely.
I was wondering whether you have examples from other environments or other gut systems where protozoa can be co-trapped without bacteria.
Do I have specific examples? No, I'm not qualified to speak on that, only that it has been done. And Jeff has a comment. I will point out that protozoa are lower in continuous cultures, but more physiological when you consider that the cultures are more diluted with buffer to maintain pH, and so probably also more diluted, not as much as protozoa are bacteria and methanogens. Yeah. So one strength of the continuous cultures is that this is more physiological. I have to remember the calculation that I did, but you can feed protozoa in continuous cultures, I think 1,000 more feed than in batch cultures, just because this is a more physiological system. So this is definitely a useful tool. Just kind of hard to maintain long term. Okay, well, thank you for all the questions.
Thank you. Looks like
yeah,
there are
if
no many
you
questions.
have any other questions that pop up, feel free to email me, and the links here again could be useful resources. So thank you.
Thank you, Tim, and thanks to all the speakers today. These were very nice presentation, and I hope we'll see you all back tomorrow for the next session with fungi and viruses. Thank you, everybody, for joining.